Hello, my name is Jeremy Warren, and I am a PhD student in Dr. Heather Hayenga's Vascular Mechanobiology Lab here at UT Dallas. And today I'm going to be going over our poster titled A Patient-Specific Computational Fluid Structural Agent-Based Model of Atherosclerotic Progression. Now, to give a little bit of context, atherosclerosis is the leading cause of death across all cardiovascular diseases. Although in vivo patient imaging and our understanding of the disease has gotten better with time, we still lack the ability to predict plaque rupture, and the treatment plans that are well suited for the generic disease lack a more patient-specific approach. To counter these shortcomings, we introduce our novel computational model capable of simulating atherosclerotic plaque progression and predicting the pathological outcomes in the hopes of gaining insight into the mechanobiological remodeling of the disease. Now, atherosclerosis is a progressive disease where heightened LDL concentrations and blood pressure drives the low-density lipoproteins into our artery wall. Now, the endothelial cells lining the lumen, they sense this and they're put under stress and release cytokines and other signaling proteins to attract neutrophils, lymphocytes, and monocytes for help. And these leukocytes migrate into the wall via transendothelial migration to address some of the inflammation and engulf the LDL. But from here, some of the LDL-laden monocytes transform into foam cells, and as time goes by, the accumulation of foam cells and excess LDL cause an initial fatty streak to form. If left untreated, this fatty streak or initial lesion can build up and eventually burst, releasing a barrage of dead cells and plaque into the blood, which can result in other pathological cardiovascular diseases and even death. Now, to address the mechanical, fluid, and biological stimuli interactions present inside of the artery, our framework uses a trifecta of modeling techniques which include finite element analysis, computational fluid dynamics, and agent-based modeling. First, we use a patient-specific intravascular ultrasound and angiogram data to reconstruct a three-dimensional artery. Now, this mesh gets passed into our framework where we use finite elements analysis, computational fluid dynamics, and agent-based modeling to simulate how the plaque growth progresses. We use FE-Bio, which is an open source FEA software, to model the internal stresses and strains inside of the artery wall. The CFD code, which was written by Dr. Stefano Leonardi here at UT Dallas, computes the wall shear stress values and the migration rates of leukocytes and LDL into the wall. Our agent-based model is implemented in Repass, another well-documented open source software, and uses a set of rules which we've derived from literature as well as the values from the FEA and the CFD to further model how the mechanobiological remodeling occurs. Now, our preliminary tests have shown that the strains and stresses from the FEA lie inside of the acceptable region, which is noted in literature and other related works. Our CFD can accurately compute the leukocyte capture rates and the wall shear stress values, and our agent-based model is capable of correctly assigning agents using the intravascular ultrasound data. Now, this said, there's still plenty of work to be done on the project. We would like to validate our outcomes and tune or optimize certain parts of the framework, and eventually we would like to study the effects that different treatments have on the disease using our framework. And some of these treatments would include pharmaceuticals, drugs, dieting, exercise, and stents. Now, as our work continues and the project moves forward, we only hope to continue to push the boundaries of what our framework is capable of and truly redefine how atherosclerosis is treated for millions of people across the world. Thank you very much.